guys, welcome back to my lectures Microbiology by Masira. In today's lecture we are going to see sterilization and disinfection techniques in microbiology. These two techniques are very important or essential in microbiology because they are needed before the start of experiments or before the start of lab experiments. So let's see one by one. Sterilization is nothing but the process by which article, medium or any type of surface is made free from microorganism either in vegetative or spore form. That means microorganisms are present in vegetative as well as spore form. So this process can remove the microorganisms from both forms through the article, medium or surface. Disinfection is nothing but the process by which pathogens get killed or disrupted, or you can say destruction of pathogen is done via disinfection technique, but it not necessarily remove spores. This is the flow sheet of sterilization. Sterilization can be done via three different methods, that is mechanical method, physical method and chemical method. Mechanical method includes different types, that is scrubbing, filtration and sedimentation. Scrubbing nothing but the using soap and water. You can scrub your hands or anything by washing. You can sterilize your equipments. Then filtration we will see in afterwards slide. Sedimentation is nothing but the water filtration or water treatment process where heavy materials or heavy flocks settle down and required filtrate is remains upside. And physical method, there are different types: moist heat, dry heat, sunlight and radiations. And chemical methods include or use or utilizes different types of liquids or chemical agents. Now we will see one by one. Filtration. Filtration is nothing but the process by which required filtrate is taken out through the filtration apparatus. According to the size of the bacteria, virus or fungi or any types of microorganism, filters are made, and through these filters filtration is done. You can see in the first diagram here shown, larger particles are settled above the filter and required smaller particles get through the filter, and we will collect them in a filtrate. In second diagram you can see test samples are poured, or you can say filtered through the filter membrane, or you can say filter paper, and microbes are trapped on the filter paper so that sterilization can be done and filtrate is taken out containing no microorganism in that. Now there are different types of filters are used during the filtration, that is depth filter and membrane filter. Here you can see in the diagram, this is the filter apparatus generally used to filter the sample. So depth filters or depth filtration means the filtration process where particles remain inside the filter instead on the surface. That means particles remain on the remain inside the filter paper you can say, and filtrate we can taken out, and they are made up of ceramic materials and sintered glass. You can see in the diagram, first diagram, depth filtration. Then second is membrane filtration or surface filtration also we can say. Then it is made up of polymers, and it acts as a siever, or it sieves the particle according to the pore size, as I have said in the previous slide. That is according to the or depending on the size of the bacteria, virus, fungi or any type of microorganism, filter papers are made. Okay, so depending upon the size, according to the pore size, the required liquid sample you can say sterilized in the filtrate flask, and not necessary requiring microorganism get trapped on the filter paper. And this filter apparatus is used in bacteriophage isolation, or you can say virus isolation, where depending upon the size of the virus, filter papers are made, and using that filter paper we can isolate bacteriophage and sterilize our sample also. Then coming to the physical methods include moist heat, dry heat we have seen. So moist heat include autoclave. Autoclave is also called as steam under pressure. In your laboratory you use you will see this type of pressure cooker where you sterilize your equipments or sterilize your glass equipments, you can say. And it involves specific time, temperature and pressure. So it uses high pressure steam to eliminate bacteria, viruses, fungi from the equipments. Okay, and this sterilization requires different parameters, as I have said: steam, pressure, temperature and time. It destroyed microorganism by irreversible coagulation. That means it coagulates the protein and denaturate the enzymes and structural proteins, so that microorganism cannot survive and gets die. Then steam sterilization is generally carried out at temperature between 121 degrees Celsius at specific time period, 10 to 60 minutes. The steam under pressure cycles carried out. So at this time period you have to carry out the cycle so that the sterilization can be done and all equipments can get removed. Now pasteurization is mostly used in the industries, where we have to increase the shelf life of the products like fruit juices and milks, etc. But most probably milk is get sterilized, or you can say the shelf life of product milk is increased via pasteurization. And pasteurization is of different types, that is ultra pasteurization, high temperature pasteurization, low temperature pasteurization, and at a specific temperature pasteurization can be carried out. And during the food preservation and packaging of foods, pasteurization can be done. This name is carried or named after the scientist Louis Pasteur. Now dry heat is of two types: incineration and flaming. Okay, so incineration is nothing but the burning of the required product, or you can say any waste material you have to burn or you have to discard, you can incinerate. In our past history there is the myth of burning of dead bodies, like so animal carcasses, soil dressings, beddings and any pathological materials which we have to discard, they gets burned via incineration process. And using the combustion process, the waste is reduced up to 70%. Then second one is the flaming is nothing but we use daily in our laboratory. That means we flame or we flame our glass tubes, or you can say nichrome wire loop and needles and etc through the burner prior to the experiments. Now ionization. Coming to the ionization is nothing but the use of radiation or electromagnetic waves to sterilize the equipments. So there are two types: ionizing radiation and non-ionizing radiation. Ionizing is nothing but the high frequency electromagnetic waves are used to destroy molecules, for example X-rays and gamma rays. And non-ionizing means low frequency electromagnetic waves are used to destroy electromagnetic waves, to destroy the molecules via electromagnetic waves, for example UV rays. Because why we use ionization? Because microorganisms are also made up of molecules, atoms and proteins. So through this radiation, all these atoms, molecules and proteins get destroyed or degenerated, so that microorganism cannot be survived, thus killed. Now coming to the last, chemical method. Chemical method are generally used during the disinfection. They are of two types: gaseous and liquid. So different types of gases can be used. They should be bactericidal, and mostly you should understand that whenever we are going to use chemicals, you must handle them very carefully and properly, because they can be toxic or lethal. So different types of gases like ethylene oxide, formaldehyde, you can say formalin, and liquids like 70% ethanol, dyes, halogen and phenols can be used during the chemical method of sterilization. And what is the mechanism of action? It coagulates the protein, it removes the free sulfhydryl groups, then they interfere in the substrate competition during the enzyme production. And disruption of cell membrane: by disrupting the cell membrane, my chemicals are interfered during their production or their growth, so that microorganisms can be killed. and thus sterilization can be done. So these are all the different types of methods of sterilization we have seen in this lecture. Thank you.